Hey everybody, welcome back to it. I hope you guys are all healthy and ready to go here with our biology class. And we'll get started here with some new concepts. We're going to kind of piggyback on what we went through with DNA before we left school and also with cell division, right? So we're going to be looking at genetics. This will be our next unit. And we're going to start off with the concept of meiosis. This will be chapter 11. I know we don't have our textbooks, but if we ever get a chance to kind of get back and take a look at those, we're looking at 11-4. And again, we're going to move on with genetics. So meiosis being the first concept that we need to go through. Sounds a lot like mitosis. Mitosis being the division of somatic cells. So there's two different types of cells. Somatic cells being regular kind of everyday cells that you're familiar with as far as skin cells, cheek cells, hair cells. All the organs are made of these different cells, so those are somatic cells, and then gametes are going to be the sex cells. Okay, so these are going to be the reproductive cells, sperm and egg, and we'll talk about how that relates to genetics. So with that, we know that parents can produce many types of offspring and families are going to have specific resemblances, but no two are going to be exactly alike. So even identical twins are going to be having differences, and those differences are going to be due to how genetics are passed on from parents to offspring. The next slide here is going to go through what we've already kind of gone through with chromosomes, and we're going to look at how the number of chromosomes in each species is different. So if we start at this first idea here, this first diagram with the chimpanzee and the potato, oddly enough, they have the same amount of chromosomes as 48 chromosomes, but we do know that those chromosomes are different, right? So obviously they have to be quite a bit different, and we'll talk about chromosomes again being packaged DNA, and all those traits are found within DNA, right? The specific codes of A, C, C's, and G's, and how that's going to be different throughout different species. So humans have 46 chromosomes, 23 pairs. Chickens have 78, frogs with 26, and then our last example, pea plants, that we're going to be talking about more of in the future when we go through Mendelian genetics and modern genetics, and we'll look at the different traits of pea plants. But again, pretty simple with 14 chromosomes total. So again, just to review the idea of chromosomes and how genes are located on chromosomes, right? Genes are going to control the traits of the individual. And if we look at the diagram, you can find chromosomes within the nucleus of the cell. We've talked about humans having 23 pairs, so 46 total chromosomes. And if we unwind each individual chromosome, we're looking at packaged DNA, right? So our DNA is going to be bound up. It's going to be wound into these structures known as chromosomes, and on the DNA we can find specific traits, right? So each specific trait is going to relate to a gene, and that's how that's going to be determined. So a couple terms that you need to know being genotype and phenotype. So genotype is going to determine phenotype. Genotype meaning genetic combinations, and then phenotype is going to be physical characteristics. So the genes will determine the physical characteristics. Now this slide's looking at a picture of the chromosomes, and we call this a karyotype. So chromosomes come in matching sets, and these are called homologous chromosomes. So again, you can see the picture of all the different chromosomes, 23 pairs. We have the sex chromosomes, X and Y, the bottom right. And it's pretty interesting that all the chromosomes are in different shapes and sizes, but for the most part, you can see how the banding patterns will line up, and that's how you can match chromosomes within this karyotype. So cells in your body have a complete set of 46 chromosomes, right? Somatic cells, 23 pairs. And a term that you guys need to know is diploid. So diploid meaning two, or di meaning two, and that's referring to two sets, right? So two pairs or two sets of 23 chromosomes is going to equal 46 total chromosomes, and cells with 46 are known as diploid. Uh, we're going to look at sex cells in particular. All right, so these are the cells that meiosis will produce, and sex cells, being sperm and egg cells, also known as gametes, only have half or 23 chromosomes, and they are called haploid. So we'll go through the process on how these haploid cells are created, and you'll see how they're split up, and we only end up with half the amount of chromosomes in each one of these cells. And this diagram kind of gives you an idea of what each of those cells look like. We have a diploid cell on the left and then haploid cells on the right. So a diploid germ cell is going to end up creating our haploid cells. In this case, we have a haploid sperm cell on the right. Again, it only has half the amount of chromosomes as the human diploid cell on the left. So if the diploid cell on the left has 46 total chromosomes, again 23 pairs, the haploid cell on the right only has 23 total chromosomes. So back to our karyotype here, little exercise and activity that you can do. We have chromosomes on the right that have specific banding patterns, and your job is to find the homolog. So remember, homologous chromosomes are identical chromosomes, and they match up, they pair together. So see if you can take a minute here. You can pause the video and match up the chromosomes. So start with the one at the top right and match it up with one of the with its pair there within the karyotype. Then you can go to the chromosome in the middle on the right, and then the one at the bottom on the right, and see if you can find the exact banding patterns that go with each chromosome. So when we look at the life cycle of an organism, of any organism, in this case the diagram is showing the life cycle of fish. We can start out with the adults, and we know that they have diploid cells. So diploid is going to be symbolized by 2 and a capital N. So that's where you can see next to the adult fish in the diagram, you have a 2 and a capital N. That means diploid, right? So it has 2 pairs of however many chromosomes that it has. So again, in humans, it would be 23 pairs, 46 total chromosomes. 46 would be the diploid number, and 23 would be the haploid number. So through meiosis, we have gametes that are formed. These are going to be our sex cells, and specifically sperm cells created by males and egg cells created by females. And when sperm fertilizes an egg, we get a zygote. Now, the gametes themselves individually are going to be haploid, right? So they're split, and we'll talk about that through the process of meiosis. The chromosomes are split, and again, when the sperm fertilizes the egg cell, we end up with a zygote, and each gamete will contribute its chromosomes to the zygote to create a diploid cell. And then after that, the zygote will form multiple different types of cells and will create the juvenile fish in this diagram, which again is going to stay haploid. It's going to be 2N, and then we'll have more growth through the process of mitosis, where those somatic cells will divide, and so the life cycle will continue. Interesting little note here on different species is that animals from different species cannot actually create offspring. They can't create viable offspring. And this is because the proteins on the sperm and egg cells don't match, and the number of chromosomes don't match either. Now, once in a while, they are close enough, like a horse and a donkey can produce a mule, but the mule is sterile. So the mule itself cannot reproduce, and it is not considered a separate species. So the definition of species are animals that are close enough in chromosome number, and again, the proteins on the sperm and the egg will be close enough. They can match to where those organisms can create viable offspring, and viable meaning that the offspring can reproduce and keep the line going. So we can't have a species unless that happens. So when sperm and egg meet, their chromosomes must match for the zygote to develop properly, and that is really the definition of a species. All right, so go ahead and take a minute to review. You can look at number 1, 2, 3, 4, and 5 and see if you can fill in the blanks. And again, if any questions arise on this, you're always welcome to write that out in an email or a chat this week and let me know if you're confused on something. So now we'll go ahead and look at the process of creating a gamete, which we know are sex cells, being sperm and egg, and the process is called meiosis. So you can see on the right, the diagram is showing the difference between mitosis and meiosis. So remember we went through mitosis earlier this semester. We were looking at somatic cells specifically. I believe we looked at onion root tip cells under the microscope, and we also had whitefish blastula, so a developmental stage of cells with the developing embryo within whitefish. But in this case, we're going to be talking about meiosis. So again, the difference here is shown in the diagram. A lot of similarities, but the end product is really what's going to be different. So in mitosis, we have a diploid cell that is going to, for a brief time, duplicate its chromosomes, creating a 4N cell, and then that cell will split back into diploid cells so that they are identical daughter cells to the parent. Now, here's the difference with meiosis. Meiosis undergoes the same process, but once the two diploid daughter cells have formed, those cells will again divide and create 4 haploid cells. So those cells will end up being what create our sex cells, and again, those will be haploid cells as we move forward. Here's a video that kind of shows the process. I'm not too sure if this is actually going to play when I make the complete video of the slideshow, so I will provide the link that you can click on separately on Classroom and watch the process that is given by the Amoeba sisters. Within the realm of meiosis, there are two processes to create each cell. So each gamete is going to have its own process, but again, meiosis is going to stay the same. We start with the parent cell. It divides into 2 daughter cells, and those cells divide again into 4 daughter cells. So the parent cells will be diploid. The daughter cells are now haploid. With egg cells, you can see that we have one functional egg cell known as an oocyte, and then 3 polar bodies are produced. And these polar bodies are going to be non-functional. The polar bodies are reabsorbed by the female, and then the functional egg cell, the oocyte, is going to be stored. So this is the process that creates egg cells that is called oogenesis, and it's a little bit different than sperm cells that we'll see on the next slide. Sperm cells are made by the process called spermatogenesis, and it's very similar to what we just saw when we were creating egg cells, in that we have a parent cell that is diploid, creates 2 daughter cells that are also diploid, and then we have um, the four sperm cells, the 4 end product daughter cells that are going to be haploid, and they will all be functional. Now, this is an interesting and important concept that we need to be aware of, and this concept will happen in prophase 1 of meiosis. So some of the video tutorials that you'll see and some of the activities that you do are going to go through all the different phases of meiosis. And again, it's very similar to the phases of mitosis, but it actually happens twice. So homologous pairs will form, and chromosomes will actually trade genes during prophase 1 of meiosis. When those chromosomes trade genes, this is known as crossing over. So you can see in the diagram some of the blue will cross over with the pink chromosome. And again, remember, chromosomes are packaged DNA, so whenever those genes are swapped, it's going to be a mixing and a so-called shuffling of the genetic deck. And whenever those chromosomes are separated in anaphase, they will go to separate cells, and this will kind of help with genetic variation. So crossing over increases the number of possible gene combinations. I just mentioned that that is beneficial for genetic variation, and genetic variation is linked to really the success of all of life. If everything had the exact same gene combinations and we didn't have any differences, number one, we'd all look the same and we wouldn't have our own unique features, and that'd be pretty boring. But number two, when there's some sort of an outside threat or a disturbance, maybe in an environment, or we have a virus that happens to threaten a species, some of those species are going to have enough genetic variation to withstand those threats and maybe be immune to a certain extent of those threats and ensure the survival of a species. So, this is one thing that's being researched, especially now during our coronavirus pandemic, to see if there are, in fact, people that have genetic combinations that will allow genetic resistance, and maybe that can be used for therapies and future medicines and drugs and whatnot to, help other people that maybe aren't so immune. So genetic variation is very important, and it's thought to be really one of the key reasons that life on Earth has been this successful and is so diverse. Again, take a minute to review on this slide. Go ahead and answer the question. Number one, the purpose of meiosis is to what? And then number two, meiosis only occurs in um, what type of cells? And again, take a minute to go ahead and answer that. And this slide is just going to recap and relate back to what we looked at on the first slide to start this presentation. Question was thrown out, what do these four puppies have to do with meiosis? And obviously we're looking at reproduction, where a parent has passed on genetic information. The process of meiosis occurred to create gametes. Gametes combined to create a zygote. The zygote grew from a haploid cell to a diploid cell, and we have our functioning organism. Last slide here. If this tutorial doesn't show up on this video, I'll go ahead and link it to Classroom so that you can take a look at it and you can get an idea of all the steps and all the phases that happen in meiosis. So take some time, spend some time with meiosis. I'll post some resources and whatnot, but it's very important before we get started here on our unit in genetics. And now we kind of see how those genes are passed and shuffled on. And again, there's going to be some more information on that before we really dive into it. So if you have any questions, let me know. My access time is 10 to 2, and I'll be available every day. So take a look at this whenever you get a chance, and do it a couple times if you need to. I would take some notes on it if you haven't already, and we should be good to go. So again, let me know if you have questions.